First of all, why I am choosing this article? Because of the chitosan have the wide variety of advantages. They are the biocationic polymer, so which is degradable and non-toxic and more compatible with other synthetic and natural as well as in the semi-synthetic polymers. And zinc oxide, which is have the antimicrobial and potent wound dressing, wound healing activity. And then the this chitosan and the zinc oxide, which is I am choosable for my project work for the wound heal enhancement of the wound healing. And next one is a introduction. This is an introduction about the chitosan and zinc and silver. The chitosan is a biocationic polymer composed of a N-acetyl glucosamine linkage. It's degradable and non-toxic and retain the moisture environment in the wound area because of their biopolymer. And then the chitosan is edible to form many of the formulations, like in the wound dressing applications and antimicrobial agents also. And the zinc oxide, many of the researchers are focused developing of the antimicrobial agents against the multi-drug resistance. This is one of the difficult one. When the zinc oxide applied all only on the wound area, the drug resistance is not curable. So we have to enhance the antimicrobial nature as well as in the wound healing effect. So we have to add some more elements within it. By the way, the silver nanoparticles could be incorporated with the zinc oxide. Um, then we achieve the antimicrobial and as well as in the wound healing activity because of their interaction between the silver and zinc oxide. In this study, we will study briefly discuss about silver zinc oxide loaded chitosan dressing for antimicrobial and wound dressing applications. This is some of sources of chitosan: Aspergillus niger, squid pen. From this all these biosources, we have obtained the chitin. From this chitin, we have undergone the deacetylation process. We getting the chitosan. Materials and methods the authors are used for these projects. The materials, the chitosan and pharmaceutical grade of the zinc oxide, silver nitrate, and the MTT, acetic acid, and the absolute ethanol. This the structure is the methanol will be shown, and the dimethyl sulfoxide DMSO, and some of the bacterial strains are required for these experiments. First one of the preparation of silver zinc oxide nanocomposite. They were prepared by the method of the deposition precipitation method. First of all, they prepare the zinc oxide solution and the respectively silver nitrate solution. Then mixed this both and stirred at 8 degrees Celsius for 4 hours. After the stirring, they cool down to the room temperature. Then the centrifuged and washed with the alcohol and deionized water for 5 times. Then dried in a vacuum oven at 60 degrees Celsius overnight for obtain of zinc and silver nanocomposite. After the preparation over of silver and zinc oxide composite, then we move to the preparation of the chitosan. We have to incorporate the prepared zinc oxide and silver composite into the chitosan. For that, we have dissolved the chitosan into acetic acid. Acetic acid is a suitable solvent for the chitosan. After the solution getting, we have to mix the silver and the zinc oxide into the chitosan solution and stir it well. Then lyophilization, then go for the lyophilization process at 60 degrees Celsius. After the lyophilization process over, we have to dry the prepared dressing. By the way, the freeze dryer is used for that. Then we have obtained the primary chitosan sponge. Then the prepared sponge is needed to different formulations, preparation of the different formulations. We have to immerse it in the immersed in the sponge into the 0.1, 0.2, 0.5 and 1.0 mg per ml of zinc and zinc oxide solution respectively under the sonication for 1 hour. After the sonication process over, we have to again back to the lyophilization process at the 60 degrees Celsius and drying process. Then when we get the chitosan silver zinc oxide composite of 0.1, 0.2, 0.5 and 1.0 formulation respectively. And some of the characterizations of needed for the evaluation of the prepared sponge is a good one or whether it is a good or not. The SEM analysis, TEM, XRD, EDX, EDX energy dispersive spectra, and the porosity measurement, swelling ratio, and the moisture retention time, in vitro antibacterial, in vitro cytotoxicity study, whole blood clotting study, hemolysis assay, in vivo animal experiments. First one of the porosity and morphological studies are I will explain in the result and discussion part. First, in this porosity measurement, we have to measure the porosity. How will we measure in this things? First of all, we have to pre-weigh the dressing and immerse it in the absolute ethanol. Then after the weighing, uh, before and after weighing, we have to calculate in the with the help of the equation P equal to M2 minus M1 by PV multiple 100 percentage for the percentage of calculation of porosity, swelling ratio and moisture retention time. The swelling ratio is important phenomenon because of the how much of the chitosan is absorb the water. We have needed to monitoring it, and the moisture retention time. Moisture retention time is also important because of the wound area. When the wound area is becoming moisture, then only the healing process is enhanced. So we have to the moisture retention time also we have to high amount we have the high amount the prepared sponge. In the sense, the the swelling ratio and the moisture retention time will be calculated with the formula. The degree of swelling equal to mw minus m0 by m0. And the in vitro antibacterial study. In this study, the zone of incubation is calculated by the for this study. The different strains of the drug sensitive Pseudomonas aureus, E. coli, Pseudomonas aeruginosa, and drug resistant MRSA. All the strains are cultured in the suitable culture medium and suitable cultured medium. Then we have to apply the suitable dressing on the medium. Then calculate the zone of incubation method. Zone of incubation of the dressing. The in vitro cytotoxicity study. For this study, we have to getting the requirements of the tree, MTT, DMEM, it's a Dalbacos medium for the cell culture medium, and DMSO. First of all, we have to DMEE supplement with 10% of volume by volume fetal bovine serum. All of this combination is kept in the 37 degrees Celsius and in the 5% CO2 conditions. After the maintenance over, we have the piece of sterile dressing is placed on the 24 well plate, then seeded with a 1 ml of cell suspension at a concentration of 1105 cells per milliliter. After the 40 hours of incubation, 0.5 ml of 5 milligram per ml MTT assay is added to it and then incubated for the 4 hours. After the incubation over, we have to add the 1 ml of DMSO and after then incubated for the 10 minutes. Then the cell viability is measured at 490 nanometer on microplate reader. Whole blood clotting study. In this study, the blood is collected from the human ulnar vein and stored in the anticoagulant PD vacuumator, and then add the 1 ml of blood sample with the sample and 0.1 ml of calcium chloride solution and sodium citrate. The anticoagulant is added to the suitable container. Then the calculate the procoagulant ratio with the help of the formula of T0 minus D by T0 multiple 100%. The hemolysis assay. This is the hemolysis assay. The blood testing solution was prepared. First, the blood is mixed with the 2.5 ml of 0.9% saline, and the dressing pieces of the prepared sponge were equilibrated in the required quantity of saline at 37 degrees Celsius. Then the diluted blood was added to each sample and incubated for 30 minutes. The positive and negative controls are calculated with the help of the formula. Hemolysis is equal to OD SAM minus OD negative, OD positive minus OD negative. In vivo animal experiment. This is a in vivo animal experiment. You will select the 20 healthy 20 mice. They were divided into the 4 groups. The 4 groups of the control zinc oxide ointment and chitosan silver zinc oxide, chitosan silver only composite. These 4 groups are divided, and each of groups of animals getting 7 mm of cutting of the dorsal area. After the cutting, we have to apply the prepared sponges on the specified area. The healing ratio is calculated with the help of the HR equal to S0 minus S by S0 multiple 100%. The result and the discussion is the morphological analysis of pictures. The A1 is the XRD pattern, B SEM image, and C is the TEM image, D EDX spectra. The XRD, this is the XRD pattern, the prepared from the zinc as well as in the zinc oxide composite, chitosan, zinc, and the silver composite, which have the all of these elements. These are the conformational pattern. By the way, you can see the prepared zinc oxide present in the prepared sponge, and which give the peak around 35 to 40, and silver also present, and give the peak and also in this area. The zinc oxide is also present. From this study, we have revealed, we have confirmed that all the elements are present in the prepared composite dressing. And the B images of the SEM image shows the prepared sponge shows the rod-like structures, and which have the size range from 100 to 400 nanometer. It's a size range of 100 to nanometer. The zoom image of the TEM image of sponge, and then energy dispersive spectrum. This energy dispersive spectrum, what are the elements are present in the which is helped to find out which are the elements present in the prepared sponge. By the way, which shows the silver, zinc, shows the peak around the 2 to 4 and 8 to 10, which indicate the present the elements are present in the prepared composite. And then the XRD pattern, BCDE is a SEM pattern. The XRD, which indicate XRD also the prepared sponge here with all of these elements, the zinc oxide, keystone, silver, zinc oxide 1.0% of formulation, which have the chitosan silver zinc oxide 1%, and silver zinc oxide nanorods, which can be indicated by the peaks around the 30 to 40. And the chitosan also present, which we can monitor with the help of the peaks in the spectral area. And the B, C, D, E images shows the sponges are the porous in nature because of they are interconnected closely. So the porous nature of the composite dressing is a good one. Then the porosity, swelling ratio, moisture retention of prepared dressing. The porosity of the chitosan alone shows around 93% something, 90 to 100%, in between the 90 to 100%. By the way, we can see the prepared chitosan silver zinc oxide in the different ratio. The 1.0 percentage shows the porosity around 80 to 90%, which is almost near to the porosity of 90 to 100% of chitosan. And the swelling ratio. The swelling ratio study. In this study, the different colors are indicated, the different formulations, and the white one is indicated, the blank. In this, all the in this 4-bar graphs, the day 1, day 3, day 5, day 7 respectively. The all these days, the chitosan have the very constant 25 to 30 around. In this, the prepared 0.1% of chitosan, silver, and zinc oxide composite shows the swelling ratio around 20 to 25%, nearly to the chitosan alone. And the moisture retention time. In this moisture retention time, the prepared sponge and the prepared dressing shows about the 30 to 14 days about moisture retention. The moisture, they protect the environment from the moisture. They can improve the moisture content in the wound area. In vitro antibacterial activity, cytotoxicity, and chemocompatibility. In this study, the different zone of incubation method. In this study, the zone of incubation method, the different strains of bacteria are cultured in the Petri plate, the suitable medium. After the culture of different strains, we have to place the disc of our dressing, cut into small piece, and put in a suitable medium. In this 0, 1, 2, 3, 4 respectively, keystone and keystone silver zinc oxide composite in different ratio respectively. In this shows the fourth, this all the pictures of the different strains. The fourth one is shows the zone of incubation is high compared to the 0, 1, 2, 3 of the all these strains. In this study, we revealed the prepared sponge of the prepared dressing have the more compatible and having the antimicrobial activity, and the cell viability of the prepared sponge. The control 0, 1, 2, 3, 4 also respectively the prepared composite dressing in the different ratio. From this, the cell viability is less in the prepared fourth one of chitosan, silver, zinc oxide composite, which will around the near to 50 to 55 cell viability. And the evaluation of the blood clotting study. This blood clotting study indicate the above one is the time of clotting, and below one is the clotting rate in the percentage of ratio. For this blood clotting study, the fourth 5 one of is we have selected blanc, gauze, bloodjet, keystone silver, zinc oxide 0.5 formulation. From this, all of you comparing the keystone silver zinc oxide 0.5 formulation shows the clotting time. They are quickly clotting and shows the 75% of clotting rate compared than the other blanc, gauze, bloodjet, and keystone alone. This is the in vivo wound healing and antibacterial study. In vivo wound healing and antibacterial study. In this study reveals between the blanc zinc oxide ointment, keystone zinc oxide 0.5, and keystone silver zinc oxide in the 0.5 ratio. The different formulations are taken apply onto the wound in the different days 0, 3, 6, and 9. In the comparison of the 9th day of the wound, 9th day of all the prepared all the others, blanc zinc oxide ointment, keystone, and keystone zinc oxide. The prepared keystone zinc and silver composite have the more wound healing activity than the other. And then the wound area closer compared to the other blanc zinc oxide and the keystone silver that the prepared keystone silver zinc oxide 0.1 0.1, 0.5 formulations have the more wound closure area, which near to 92, in between the 90 to 100%. And this is the colony counting method. In this study, we have to collect the human after the 3 days of the surgery. We have to exudate the culture the exudate on the wound area. Then culture is needed to isolate into petri plate the suitable media. After the media is cultured, we have to chitosan. Then the prepared dressing is applied onto it. From this study, we have to conclude in the 3 and 5th day. 3 and 5th day, the preparation of the prepared keystone silver and zinc oxide 0.5 formulation shows the more less colony formation on the plate. In the histopathological evaluation, which have differentiated from H&E and PSR staining. The H&E indicated the hemolytic eosin, and the PSR is the something Picroceras red. And from the blank zinc oxide ointment, keystone zinc oxide, and the keystone silver zinc oxide preparation. In this all, we have to compare the last one of the keystone silver. This one is better wound closer compared than the blank and zinc oxide of the H&E and PSR staining also. Because of the reepithelialization is not done properly in the blank and zinc oxide ointment. From this study, we have to conclude the prepared keystone and prepared dressing is suitable for the wound healing. And the dark spots are indicated the tissue granulation formation around the area of the closing wound area closing. And the PSR staining of 5 formulations are indicated. The red color is different from the keystone silver zinc oxide and these 4 of. From this study, we have concluded the collagen formation is higher in the prepared dressing applied area. In the conclusion of the study, in this study, the prepared silver zinc oxide loaded chitosan composite sponge shows the more potent porosity and swelling ratio, as well as in the moisture retention time around the 30 to 14 days, which all the results are shows the potent wound healing and antimicrobial property. From this study, we have to demonstrate that the prepared keystone silver zinc oxide composite dressing have a potential application for the wound care and antibacterial study also. So the references I have referred for the preparation of this article. Thank you. Thank you for